We're doomed, we're saved.
The Biorevolution podcast.
Your hosts, Louise von Stechow
and Andreas Horchler. We're doomed, we're saved, episode 22. We called it Polygenic Risk Scores, the Truth in Our Genes. Welcome, as always, from daughter Louise von Stechow, biology, PhD, and father Andreas Horchler, a journalist. Polygenic risk scores, put very simply, look at gene variants across the human genome in order to determine an individual's risk of getting a disease. So in the next 30 minutes or so, we'll try to take a closer look at recent developments in PRS, short form, for polygenic risk scores, and the promise of better treatments through early predictions, as well as the potential risks. For instance, new biases that are possible. But easy, first things first, you brought guests and you brought quotes.
I will start with the quotes and then I will introduce our guests that I'm really happy are on our episode today. So for this polygenic risk scores, as you just introduced, it's a way to assess an individual's risk of developing a certain disease. And the question is, of course, also, when you are a doctor, how would you communicate this risk to your patient? And also as a patient, what could you do with this information? And I brought two quotes that actually relate to that. One is from Ali Turkmani, who's a geneticist at the Scripps Research Institute in Läll, who says, effectively communicating that a high risk score does not mean someone will definitely develop a condition, and that a low score does not mean that they will remain disease-free, is key to driving the adoption of healthy behaviors, which I think sums up a lot of the discussion
Absolutely.
that one could have on the topic. The other one is also an avenue which is actually important to look at, which is the question, would you want to know the risk of developing a disease for which there is currently no intervention? And here we have a quote from Rahul Desikhan, who was the Dean of Precision Neuroimaging at a university in San Francisco. And he was actually diagnosed with a very aggressive form of ALS, and he's actually deceased since. But he wrote in Stat magazine an article about polygenic risk scores and about knowing disease risks. And what he said, I would have wanted to know what was headed my way so I could start preparing for it and make every hour count, which I think is also in perspective on such a topic that, can be taken into account. So now I would like to introduce our two guests, Brooke and Kristi. They're both researchers working at the Intervene project, and this is a really cool interdisciplinary project that tries to make sense of how polygenic risk scores can be developed, but also how they can be used in a beneficial way for a large part of society. And maybe I can hand over to Kristi for introductions, a little bit about your background, and maybe also a little bit about Intervene, and then to Brooke.
Okay, hi everyone. My name is Kristi. I work in University of Tartu in the Institute of Genomics. I have a background in statistics and I've been working with data, including genetics, or mainly focusing my research on polygenic risk scores for the past 10 years. And the Intervene project was something which I really enjoyed working on because it pulled together data from 7 large cohorts or biobanks, and it allowed us to harness the power of polygenic risk score analysis to the max, I think. And we found out many different things about polygenic risk scores we did not know before. For instance, that the predictability depended on sex or age. So it was really, really interesting for me.
Very nice. And maybe I can hand over to Brooke for introductions.
Yeah, hi, I'm Brooke Wolford. Really happy to be here. I'm a postdoctoral fellow at the Norwegian University of Science and Technology. I'm actually originally from the US, and I'm here in Norway because they have an amazing biobank that was part of Intervene. That's the Trøndelag Health Study. So my background is in biology, statistics, and bioinformatics. I would say I'm a statistical geneticist. So I've worked on genome-wide association studies before I started working on polygenic risk scores. And the GWAS are sort of the backbone of the polygenic risk scores. So I've sort of made my way into this risk prediction field. And
Welcome, guys, from my side also. Being surrounded by specialists to such an amount is fantastic for a podcast like that. Let us try and rewind for a second, because roughly 20 years ago, in the euphoria of decoding the human genome, word spread that, well, now it would only take a small amount of time to diagnose and to cure all sorts of diseases. Didn't happen. Why?
Yeah, good question. I think so back then, I think the euphoria was really high, and I think Clinton,
It was.
Yeah, it was President Clinton who said that this is the most important map that man ever created. So, I mean, I think the idea was now we know what's in the genes, which, I mean, is also like, I mean, decoding, it was not the genome of every person that was decoded, but of a couple of people. But knowing what's in the genes will tell us how to treat any disease, which did not transpire yet. So part of that, of course, is that we don't completely understand how much of the risk of developing a disease is genetic, but certainly a part of a disease risk is genetic. There are some so-called monogenic diseases, for example, cystic fibrosis, Huntington's disease, sickle cell anemia, for which there is one gene, and it's clear that you have a risk to develop or certainty to develop a disease if you have a mutation in this gene. Many other diseases, which I think were the ones that people were thinking about in line with the Human Genome Project, like, for example, metabolic diseases such as type 2 diabetes, different types of cancer, neurodegenerative diseases such as Alzheimer's or Parkinson's, have a strong genetic component, but it's not as simple. So there's mostly, I mean, in some cases, there is also this one gene which can lead to the disease, but in many cases, there is not. So there you have multiple variants spread over the genome, which lead to this disease risk. And maybe I can hand over to our experts to explain that a little bit.
Yeah, so I can try to take that one. We have found that there's just so much more complexity in the genome. There's lots of variation between you and me that we have no idea what it does. Could be completely benign, just things that have cropped up over generations of humans. There's sort of this polygenic model, that this idea that all these variants across the genome each have some small effect. We often think in terms of additive effects, but there's also multiplicative effects and feedback loops and all sorts of interactions that are hard to model appropriately. And I mean, there's even been some talk in the last couple of years of like an omnigenic model, which is like every variant in your genome is somehow very slightly affecting some like core groups of genes for a given trait. So it's definitely more complex than scientists originally thought.
Are we getting closer to actually harnessing this instrument of understanding polygenic risk in terms of predicting a certain disease risk?
I would say that we are getting closer to making some sense of the data we have. We are definitely not close to explaining all the heritability for all of the diseases, but we can do pretty well for some of them, that we can take what we know and we can make use of it, which I think is very important. That there has been, if I'm not mistaken, like almost 20 years of GWASs. So it's great that we can make sense using them for at least some of the diseases. But I don't think we are near anywhere explaining all of the genetic background of the diseases.
Getting closer, to say the least. I would be very quickly interested in how you organize your work in such a European circumstance. I mean, you both are very different places, and I assume that other scientists are other places as well. So how do you get together and share your insights and your findings?
Yeah, so the Intervene project is an EU-funded project. I don't even remember how many partners there are, but there's like a core set of biobanks that signed on saying we want to be a part of this. And all the data stays in the individual countries where that biobank is. So we have analysts that sort of represent each biobank. We work together to come up with code and we share it to each of the biobanks. They run the code, work out any quirks of their specific data set, and then share summary statistics back, and then kind of a core group of us analyze together. We have phone calls every Friday to discuss. We've had several face-to-face meetings. There's this dream of having like federated data access, but data privacy and GDPR and a lot of things come into play. So for now, this is how we manage keeping the data in each country where it belongs.
Easy, back to PRS and to the traditional way of predicting disease risks. Very obvious. I mean, if you're obese, the likelihood of you acquiring diabetes grows. If you smoke a pack a day, your risk of lung cancer and cardiovascular disease grows exponentially. But at that point, we're not talking that much about genes, but more about behavior. And my understanding is we're facing a mix of very many factors here, but the traditional approach to get to our core topic today, how would you explain that real quick?
Yeah, I mean, in general, I think there are, of course, for many different diseases, different risk factors, but for general screening purposes, often, I mean, for example, disease risk increases with age. So over a certain age, you might be screened for different types of cancer, colon cancer, and the likes. For example, that starts at 40, 50, 60. Maybe there you would start screening for prostate cancer. So this is one important factor. Then, of course, as you said, your weight or your BMI is an important risk factor for metabolic disease. And then there are, of course, the family history. So there are some, of course, inheritable diseases. So there, if you, for example, have a close relative with inheritable form of breast cancer, for example, with a BRCA mutation, you, of course, have an increased risk of also developing this, and this will also be taken into account. And then, of course, there are a lot of screenings of laboratory biomarkers or even preventative biopsies or checking if you might be developing something right now. But all of that is currently not, I mean, it really depends also on the healthcare system of the country you're in. I'm speaking for the German system now. I think this depends very much where you're located. But most of that is not very targeted towards the individual. So this is a little bit of a one-size-fits-all approach we talked in 2 episodes ago a lot about women's health versus men's health, or in general, like having more specific measures for what well-being and health means for a woman as compared to a man. And this, of course, has this, let's say, relatively broad component, and in many cases doesn't take into account the genetic risk. And of course, the genetic risk is present since your conception, so it's present since you were basically in existence. And this is, of course, quite interesting because it allows much earlier to determine if someone has a likelihood of developing a certain disease and then intervening, potentially.
Yeah. Maybe our 2 experts can, now that PRS entered the scene, so to speak, and is a very young discipline, talk a little bit about percentages. How close do you get at the end of the day to predict, okay, you are running into the risk of developing diabetes or a certain form of cancer or something?
My take on it is that we cannot estimate how precisely polygenic risk score alone estimates your disease risk, as I think very often polygenic risk scores, at least among medical experts, is that it should be treated as a diagnostic tool, which is, I don't think, the right way to think of it. I would think of polygenic risk score just as one variable which helps the absolute risk algorithms to become way more accurate. Like it's a similar type of risk factor as a BMI or your family history or something else. You add it in the risk model and it helps the risk model estimate to be way more precise. And just to give a simple example, many people know that these BRCA mutations are very important for breast cancer risk estimation. But it has been shown, I think, a couple of years ago that when you include polygenic risk score also in the algorithm, then you can get way more accurate risks for who gets the breast cancer, even in the presence of the BRCA mutation. So combining all of the information we know will get us the best result, is what I would say.
So huge progress in the making as we speak, no doubt. And obviously there's a huge potential in PRS. But at this point, we're met with a translational aspect of things that seems to be present in many fields within the healthcare sector. So how and to which end will we implement PRS into the clinic if at all, and when?
Yeah, that's
many questions in one.
Of course. Yeah, I think this is a really good question. And I think in general for including new instruments into clinical practice, it's of course always a challenge of how to deal with it. But I think in general, the question here is, and this is a question that's basically unanswerable, but maybe we need to approach it to a certain extent, is to find a way where the benefit outweighs the risk of implementing a new tool. So you want to have it as precise as possible, and you don't want the people who, it's very, clear, for example, for medication, you do clinical studies to test, does the benefit of giving this drug outweigh the adverse reactions of having that? There is also the other component of what does it cost, and for how many people can it be beneficial. That gets even more difficult to entangle or disentangle. But I think here just the question of finding a way to say, okay, this is on the one hand good enough from the predictive prowess that it has, and it can help, as Kristi just said, to complement other risk scores, but also how can physicians really implement that in a way that they communicate it to their patients that, we had it with a quote in the beginning, we're not talking about an absolute risk. We're not talking about you will for sure with 70 years develop this disease, but it's a question of relative risks, which is not so easy, I think, to communicate. And maybe I can hand over to Brooke for elaborating a little bit on that.
Yeah, so one of the work packages in Intervene is actually looking at some clinical trial to try to understand how can we implement this, translate it into the clinic, how does it improve upon existing clinical risk measures. I would say the three diseases where there seem to be pretty direct applicability to either lifestyle interventions, medications, screening programs are heart disease, type 2 diabetes, and breast cancer. And there's clinical trials going on around the world right now for some of these. I know breast cancer, there's a really nice study in the UK that will hopefully answer some of these questions. And I think Kristi has some experience from Estonia actually for breast cancer, for PRS being implemented, if maybe you want to touch on that.
Yeah, so just shortly, we did a pilot a few years ago in Estonia where we invited females who had polygenic risk score among top 5% to meet with the oncologist when they met certain criteria, their age, and they were breast cancer diagnosis free and so on. And it actually turned out that people were very interested and they showed up to get the results, and they are being also followed up now later. And we did some cost-effective analysis, or some other researchers in University of Tartu did that, and they stated that it's actually cost-effective to implement screening in earlier ages, starting from 40, and maybe also extending it for high-risk individuals beyond the current age limit. As just to give a simple example, the polygenic risk score we used by the age 70, in the top 5%, about 12% of females were diagnosed with breast cancer. The population average is, I think, 6.5 or close to 7. And for example, among lowest risk score category, the percentage of females with breast cancer diagnosis was only 3%. So there is a 4-time difference in how many people get the breast cancer diagnosis by the age of 70. So exactly if 12% of women get the disease, it means that 88% do not. So like Louise said, it's not an absolute estimate that if you have high genetic risk estimate, then you get the disease. No, but you are way more likely compared to the population average or the lowest risk score groups to get the disease than the others. And that is, I think, the power of polygenic risk scores. It helps us select individuals who are way more likely to get it.
And I have to say, and this is also something that's, of course, very interesting for many of those diseases where you actually do have an intervention. Type 2 diabetes is maybe the simplest one, but where you can also do something preemptively about it. And this can be quite helpful. And I think that was very initial results, but I saw that there were some studies that showed that people were, once they got the communication of their polygenic risk score, they were actually more likely to adopt health-promoting behaviors. So they would stop smoking, exercise more, eat more healthy. And that, of course, if this would hold true in a larger scale, that would, of course, be an excellent model to know, okay, you're at a certain risk and you can actually do something about it.
Definitely. I think it's one thing I have read from the literature, I can't remember the exact title of it, was that people need reminders. For instance, that I tell you that you have high risk for something which is not, let's say, deadly or very scary for you, then you tend to forget over time. So for the behavioral changes to last, you kind of need some sort of intervention or reminder, I would say, every now and then, so that it will be in your mind, okay, I had high risk for this. I should be more motivated to do more healthy stuff for myself. So I think that this one-time intervention for majority of diseases maybe is not enough. It's something which you need constant, or at least every now and then, reminder. Maybe it is, and it's my personal opinion, I don't have any science to back it up, but I think it is more difficult for cancers, meaning if you get a result for a cancer, you are, at least I don't think that you're very likely to forget it.
And as Louise hinted, it's a lot about the communication in the clinic of the person who gives a diagnosis. And as we are talking in this podcast also, is that the drug development, even when it comes to cancer, is breathtaking what's happening right now. So even if you have a diagnosis like this, that today doesn't mean the end of hope, of course, Yeah. I would like to, because we have some things on our plate, I would love to talk about the communications even more, because this is my field of expertise, but I would love to talk about also, as we are in Europe, about validation regulation. We all know that from many examples that the EU ticks a little different from the rest of the world, namely the US, of course, when it comes to data protection. Some scientists argue that a strong regulatory framework will, at least in the long run, help to protect both standards, safety and transparency. But other things judge differently. But first off, we would need to tame the data in order to make it accessible. And I would be very interested to hear a little bit about where we stand right now.
Yeah, I think I would actually hand that over to the two of you, because you're much closer to the question of data. But I think, the question is how freely accessible and how transparent, on the one hand, can data be for scientists to use and also to compare different polygenic risk scores to one another, but also, on the other hand, to actually protect the individual's data. And I think we'll talk in the end a little bit about direct-to-consumer companies where, the picture might look a little bit different than in an academic context.
Yeah, so I'll just say, luckily, the weights files that we use to calculate a score, they're like chromosomal positions and then estimates of basically the risk that specific chromosomal position is estimated to give on a certain disease. And so that's not anyone's data specifically, that's like considered a summary statistic. And so that can be publicly shared. And there's a really nice site called the PGS catalog that they've done a really awesome job trying to make these weights open access so that people can go and validate other people's scores, try to understand how they're performing, and that there's transparency in what ancestry genetic data was used to assess performance originally, or from the GWAS that was used. And that, I think, is really important for when you actually put a score into the clinic. Like, you want to know that it's optimal, that it's working for the most people possible. And so I would say the PGS catalog has gone a long way in putting us closer towards that.
But maybe at that point, there's also a little bit, because it's called We're Doomed, We're Saved. So we need to talk a little bit about the potential doom, at least. So ancestry biases, potential biases that might be involved in the data. What are we talking about here?
I think this is a challenge, and we talked about that at length for the question of sex segregated data in the Women's Health episode. The problem is that there is a bias in these genome-wide association studies which underlie this original scores. And there is a certain bias towards men and strong bias towards people of European ancestry. So people of diverse ethnicity are often not captured. And also there's often, and Brooke and Kristi, correct me if I'm wrong, but as far as I understand it, often we don't really know what was in this data set. So basically, we do not know if it's women or men originally who were assessed in a certain study. So basically, you cannot segregate and then not make it more specific. But that might also mean that these polygenic risk scores are much more predictive for the original population on which it was assessed, which might just be men of European ancestry or people of European ancestry.
So I can chime in on the sex part. So we do, for the most part, know, okay, this biobank, this cohort study was X% female, X% male. I think from the work that Kristi and I have more recently done, one thing that would be a real strength is to have a sex stratified GWAS. So right now we kind of pull everyone together, but we do see that there can be differential effects of a certain genetic variant on men versus women. So if we had statistically powered sex stratified GWAS, I think that would actually go a long way. So in general, data is pooled, but it is well described. And there's actually, from a lot of the biobanks I've seen, there's actually a participation bias towards women in a lot of the biobanks, unless there has been historic cohorts that were maybe more male-centric. So it's a mix, I guess, I would say, in terms of that.
And in terms of the ancestry?
Yeah, so there's been a lot of effort in the last, 5 years to try to do a much better job in terms of ancestry. But in the end, it is the US and European countries that tend to have these large biobanks. And to a larger extent now, China and Japan and Qatar have some biobank efforts going on. But the GWAS that are, like I said, the backbone of these polygenic risk scores, they have just been predominantly in these countries that are predominantly European ancestry individuals. So we need efforts to build biobank infrastructure globally, which has definitely also been an area of focus. And then we need to do the best we can to use the multi-ancestry samples that are available, both for the GWAS and for evaluating the scores and trying to understand how to make the scores as globally useful as possible. Kristi might have some other things to say there about ancestry.
I think Brooke covered it real well. Like she said, there are several efforts now and a lot of attention put to it that we would take ancestry into account. For instance, I think also polygenic risk score calculation tool, which is also developed by Intervene researchers. It now, for example, adjusts or standardized for genetic ancestry, which is determined from the data so that we could adjust polygenic risk scores and make use of them for everybody. But this does, of course, not help with the fact that due to European bias in GWAS, the scores based on that data is more well-suited for European ancestry individuals, and it does not work that well for other ancestry groups. I think there are also some efforts going on. If I'm not mistaken, it's the eMERGE project, which has the entire workflow describing how polygenic risk scores should be selected for different trials or moving on. And they evaluate them in 4 different big ancestry groups. And they select them actually that way that they try to select scores when they are working for other groups than European ancestry groups to kind of make it more equal. But it's definitely something which needs a lot of attention, and we're not going to overcome this in a year or 2.
Yeah, I think one could summarize it as work in progress, but I think also an acknowledged challenge within the data, which might also actually, if this is an instrument that, would be implemented clinically, it could also actually help to boost the research towards different ancestry groups and also towards segregating data more towards different identifiers, which could be actually maybe even a helpful way to move more towards the right direction there, I could imagine.
Yeah, and I'll just chime in and say the paper that Kristi and I have worked on together the last couple of years that hopefully will be out soon, it's in the European ancestry samples of the biobanks that are part of Intervene, because when we started looking at African ancestry and East Asian and South Asian ancestry, we didn't have the statistical power to look at these age and sex stratifications that we were doing. So now a group of us is revisiting all these cohorts, and we've gotten more cohorts on board to hopefully increase our power so that we can make the same kind of how does sex and age interact with your polygenic score in populations that are not just European. So we are trying to be a part of the solution, but it's an ongoing effort to try to find the biobanks that have sufficient sample sizes in non-European populations.
We have not said anything about the limitations, maybe besides ancestry. And I think one thing I want to say, so to be transparent on the topic, is that polygenic risk scores at this moment are estimations. It's not the truth, and they are bound to change in 5, 7, whatever years. We know more, we get a better estimate. So people have to understand that it's not like the monogenic variant that you have it or no. With polygenic risk score estimate, it's an estimation. We estimate this, but it's bound to change. And also, before we implement anything in the clinic, we have to have guidelines of what happens if I today tell you that you have a high risk for polygenic risk cancer, and then in 3 years I tell you that the new estimate is that it's moderate or even low. So what happens with you? What happens with what you've been doing? Maybe you have been screened more often. Are you then going to be screened as like an average population? So these type of things, we need to figure them out before we implement these things in the clinic. And it's not something which is theoretical. It is proven already that 2 different polygenic risk scores for the same disease give different results. So we have to take this into account, and we have to tell the people it's not something which is a certain thing for the rest of your life. It might change.
So let's try to wrap up a little bit. Of course, I would be very interested to hear a few words about a vision about when will PRS be a new or a normal in itself in the healthcare system, and where is it applicable outside maybe the healthcare system, and what kind of economic models are there that will help in the development of PRS integrating?
Yeah, I think the time horizon I leave to our experts because I have no clue. But thinking about the applications outside the healthcare system, I mean, there are a number of direct-to-consumer companies that have worked with monogenic variants and are also exploring polygenic risk scores. 23andMe comes to mind. This is a very famous one. I think it's definitely a business model, and there are a lot of people who get an analysis both of their ancestry based on these genomic profiles, but also of potential risks of developing a disease. It's my very personal opinion, but I think it's great if this is within the healthcare system because it requires a statistical and a genetic literacy that is very difficult, I think, to transmit to a person who just puts in something to a computer and gets back, you have a 13% risk of developing Alzheimer's. What do you do with this information?
Nothing.
I think it's very, very helpful to do that with a doctor who can put this into perspective and also explain what relative risk means for you, instead of doing that by yourself. That said, I mean, it could be also that in the future, these direct-to-consumer companies will have such good explanations, I don't know in which format, that it's okay for the person to deal with this information by yourself. But I think in general, the healthcare system provides a safety net and also psychological safety net, and also gives you a catalog of interventions you might do for the disease risk that you have. And if you just do that all by yourself, you might run into problems that don't even exist, basically. And I think that for me is a challenge. Yeah, and maybe I can hand over for let's think about when this will be clinically applicable and how you feel maybe about the direct-to-consumer situation.
Yeah, I'm a bit torn because I do think knowledge is power, but I also recognize how anxious it could make someone to get this risk. They really don't know how to interpret. I think genetic counselors play a really big role here, at least in the US framework. I don't know how common genetic counselors are globally, but trying to help people interpret these findings. And I think we actually need more trained genetic counselors. And yeah, I think that's a tough.
I'm not even aware if we have something like that.
Yeah, we do.
In Germany, we do.
We
I
do.
never even heard the term.
No. I mean, for example, in the rare disease field, you have that.
Okay, all right.
People who communicate results once you know that.
Yeah, understood.
But it's like in specialty centers.
Okay. Yeah, and it's really existed in that Mendelian space in this world where things are more polygenic now. We need to train genetic counselors to interpret this polygenic thing, which is very different from a Mendelian framework. And then, boost a workforce that's able to communicate these things. Because I don't know about you guys, but whenever I go to a doctor, they have very limited time to talk to me and work with me. And some of these conversations are a bit longer winded, maybe than most healthcare systems are prepared to handle.
Yeah, so I have kind of two completely different ways of approaching this topic. If you want to talk about directly tests which are directly available for individuals, you can buy something on your own. Then I think one field where it definitely should not be used is this embryo selection. I find it irresponsible because we know only some part of genetics for a majority of diseases. And then we make decisions based on just that some part, which is a decision of who gets to basically at least a chance to live, no? And when we select this type of things, do you have a genetic predisposition for diabetes? You don't actually know what you also select because we don't know exactly how things are intercorrelated with each other. You might select also some other things you're not even aware of. So it's a very muddy field. And I think also, not to make a mistake, the American College of Medical Genetics and Genomics also deemed that the pre-implantation PRS testing is definitely not something we should be doing at the moment. And I agree with them. As for the feedback of diseases which we can do something about on our own, then, for example, Estonian Biobank is currently piloting genetic feedbacks for everyone. We have released the platform for a couple of thousands of individuals, but by the next year, I hope, I'm not promising, but I hope that all 200,000 participants can access it, and they will see some information about each other. And as Brooke said, these are type of conversations which you need longer time for. So it's kind of like a huge platform where you can read about the disease. Also, you can read about what to do. You can read what the genetic risk means, how it changes your absolute risk if you have a low genetic risk or high genetic risk. So you can understand a little bit that it's not a 0 or 1 decision, but it's a probability. So we will know in a few years how participants in Estonia kind of received it. There was a smaller study a few years ago, and then participants said that it's very valuable. We tested it on 3,000 people. Maybe testing is a bad word, but 3,000 people accessed the results. So, and they gave feedback, and they said they find it very valuable, and majority of them were content and not very worried. But it was with the genetic counselor. So this time it's the first time they read it on their own, and there won't be somebody to directly advise them. But I think it's very important, like Louise said, that people wouldn't be alone and like, okay, what do I do now? So we definitely need not to leave people alone with the results.
I'm hearing, guys, that you are part-time diplomats, not saying like by 2030 it's going to be standard. But in a way, I didn't even expect this because you got to be cautious. I understand that.
Yeah, Nothing to add.
I was going to chime in real quickly and say that I think this is an interesting also like barrier to research, because we want to research it and see how well does it work? Is it cost effective? Does it induce anxiety? Does it actually help save lives? But the budget that would be needed, like Kristi said, the pilot had genetic counselors, but then when you expand it, the price that would be needed to do that for all your research participants would be really high. And so I think this is, we need more funding for science. That's my call and my lesson there. But I mean, I think that there's enough people that understand how to communicate risk that these kinds of things can be done well. And I think a research setting is a little bit different from a direct-to-consumer testing. But in a perfect world, everyone would get these results with like a medical professional, you know?
I'm sure Brussels is listening right now when it comes to budget.
I hope so.
I also want to say one thing about, this cost effectiveness, which is a really difficult measure, I think, in healthcare systems. But disease prevention is always cheaper than disease, because disease costs not only treatment for the disease, but also loss of productivity. And that's very expensive. So people not getting sick is much better.
Yep.
Not only for the individual, but also for everyone.
Nice closing words, I think. All right, guys, this was We're Doomed, We're Saved, episode 22. Many, many thanks to our guests and their expertise. Of course, we're going to put links to your work to the show notes. Easy, do we know anything about moving forward? What's in the pipeline? What's planned for next episode?
We do. We will examine if the new technologies in biology, such as PRS and many others, actually would count as a new scientific revolution. So we'll dig back again a little bit in the history of science and talk about the famous book of Kuhn from the 1960s and re-examine the scientific revolution, and if it is one.
Thank you to both our guests.
Thank you very much.
Thank you.
Thank you for having us.
We're Doomed, We're Saved, episode 22. Polygenic risk scores, the truth in our genes. If you like the show, please subscribe to it at the streaming service of your choice, or visit us at sciencetales.com. That's science-tales.com for feedback, further information, and inspiration. Thanks for listening.